Hey guys, this is Miss Adelini, and this is the second part of the Enzymes and Introduction to Metabolism lecture. In the first part of our video, we talked about metabolism in general, the types of energy, and the types of reactions, endergonic versus exergonic. We also talked about how the energy from an exergonic reaction, like the hydrolysis of ATP, can be used to fuel an endergonic reaction, one that requires an input of energy. In this second part of our lecture, we're going to talk about enzymes, which can be used to speed up reactions, and talk about how they do that. An enzyme is a specific type of catalyst molecule. A catalyst is defined as anything that can change the rate of a reaction, usually speeds up a reaction, without being consumed by the reaction. In other words, the catalyst can be recycled and used again to help speed up that same reaction. An enzyme is a catalytic protein, so it's a specific type of catalyst that's a protein molecule. Usually an enzyme can be identified because its name ends in ase. So an example might be amylase, salivary amylase, which is the enzyme found in saliva that breaks down starch. Okay, let's skip down. Usually enzymes work by lowering what's called the activation energy of a reaction. And for the most part, all reactions have an activation energy, and that's defined as the amount of energy that's needed to push the reactants over a particular energy barrier so that they can be transformed into the products. If you look at the little graph down here, it shows us our reactant molecules, molecule AB and molecule CD. And we have to put some energy in order to weaken the bonds between AB and CD so that they can be recombined to create molecules AC and BD, are products of the reaction. So the energy that's put in order to weaken those bonds is called the activation energy. At the very top of their free energy curve, the molecules are in what's called a transition state. They're very unstable. AB is about to break apart, as is CD. Something that can serve as activation energy is heat. In this particular reaction, we're looking at a reaction where initially we have to add in some activation energy like heat, but overall we lose energy in the reaction. The delta G, or change in free energy, is negative. So we are overall releasing energy. It's exergonic. Okay. Though heat can be used as activation energy, there's usually not enough heat for the molecules to make it over that hump and get past the transition state. If we had enough heat to get them over that transition state, it would be so hot that it would also denature the proteins in our bodies and it could kill the cells. So what we're wondering is, how can these reactions take place if there's not really enough heat for them to overcome their activation energy barrier? They do so with the help of enzymes. Enzymes work by lowering the activation energy required. So even when we have moderate temperatures that your body's cells can survive at, we can still get over that activation energy barrier since the enzyme has lowered it. They don't change the overall delta G, or free energy change, which is the change from the reactants to the products. That's still the same. All they do is lower the activation energy. So in this graph, we see that we have our black line representing the reaction without an enzyme present. Notice it has a much higher EA, or activation energy, than the reaction shown in red, which is the same reaction, but here we have an enzyme, so our activation energy is lower. Enzymes are going to speed reactions up because they can get over that activation energy barrier quickly, and that's going to help with reactions that would occur naturally in the cell, but not fast enough to help the cell out. This is a really important graph, and I actually ask you to draw this graph in number 5 within your notes. You're asked to draw a graph to show how an enzyme-mediated reaction, which is just a reaction that's assisted by an enzyme, is different from a reaction that proceeds without the help of an enzyme. So there, I'm talking about this graph. In a reaction that's catalyzed by an enzyme, the enzyme has to fit pretty specifically with the reactant molecule or the substrate in order to facilitate the reaction. So here's a picture showing the breakdown of table sugar sucrose by the enzyme sucrase. Notice how we have ASE in the enzyme's name. Here, sucrose is considered our substrate. Substrate is a term to describe a reactant that's found in an enzyme-assisted reaction. So this is just a more specific term for a reactant. Our sucrose molecule has to fit pretty exactly within the enzyme sucrase. When they're together, we call them an enzyme-substrate complex. And then somehow, the enzyme is going to facilitate the breaking apart of glucose and fructose. In this case, notice that we're adding a water molecule. That indicates that this is the hydrolysis of sucrose, or the breaking down of sucrose using water. Now that could happen on its own, but it's going to happen much more quickly with the enzyme sucrase. Once the substrate has been broken down into our two products, glucose and fructose, it will release those products, and then our enzyme is totally unchanged by the process. It can be used again for that exact same reaction. Okay. The way that substrates match up with the enzyme are they're attracted to the active site by some sort of weak bond, like a hydrogen bond, and occasionally it would be an ionic bond. Let's say the substrate has a slight positive charge, the enzyme has regions of negative charge, they'll be attracted to each other. It also has to do with the shape of the substrate molecule fitting into the shape of the active site. The active site is just the specific region of the enzyme where the substrate is going to bind. Each enzyme has a specific shape and chemical nature of its active site so that it can only bind one particular substrate. We can have enzymes that are used to break apart one substrate into several products. We also have enzymes that assist with anabolic reactions, taking in two or more substrates and joining them together to make larger product molecules. Enzymes are going to use two different ways to lower the activation energy in order to speed up a reaction. If it's an anabolic reaction, one where we're taking in several substrates to put them together to make one product, the active site is going to orient or place the substrates in the correct position for bonds to form between them. Making bonds between those substrate molecules will create one chain or one product molecule. If it's a catabolic reaction where we're breaking apart one substrate molecule into several products, the enzyme substrate complex could put stress on the bonds within that one substrate molecule that makes it easier for them to break. Something that scientists have been debating is the way that the enzymes and substrates fit together. Initially, scientists believed that enzymes and substrates fit together like a lock and key. The enzyme is going to represent the lock with its active site being the keyhole, and then the substrate would fit in exactly into the keyhole. Now we think that the induced fit model is more likely. They think that once the substrate binds to the active site, the enzyme is going to change shape slightly. That allows it to bind the substrate more firmly, and it puts a strain on the bonds within that substrate. So that's going to refer to our discussion of catabolic reactions, where when we bind the substrate, it puts stress on those bonds as a result of the enzyme changing shape slightly to better fit the substrate. We also discuss it up here in the anabolic reactions. The shape of the enzyme will change due to induced fit. So once the two substrate molecules bind, the enzyme is going to change shape slightly to hold them better, and it's going to put them in a position to bond with each other. Because enzyme molecules can be recycled and reused, a single enzyme molecule can catalyze or help with thousands of reactions within a second. An example is an enzyme found in bacteria that are in the air. It produces an enzyme called urease, which can be used to break down a compound called urea that's found in urine. So within a kitty litter box that's going to have cat urine in it, the enzyme urease is going to assist with the breakdown of urea into carbon dioxide and ammonia. Without that urease from air bacteria, it says it would take 3 million years to break down the urea from one day of cat pee. Nasty. Okay. So in your actual notes, I skipped through this a little bit, but they do talk about activation energy, enzymes and substrates, and the active site. Okay. At this point, we're going to start talking about certain factors in the environment that can affect the efficiency of a particular enzyme. So let's take a look at temperature. Each enzyme is going to have a temperature that it functions best at. We call that the optimal temperature for the enzyme's activity. And within our bodies, the optimal temperature is going to be around 98.6 degrees Fahrenheit. Now, below that optimal temperature, if we're down here, let's say for this particular enzyme, below 30 degrees Celsius, the problem is the enzyme and the substrate are going to be moving so slowly at that low temperature that they rarely collide and find one another. So the rate of reaction is going to be lower simply because the enzyme and the substrate don't meet up frequently. As we raise the temperature past this 30 degrees and up to 40, the rate of reaction will increase because the molecules, the enzymes and the substrates, are moving more quickly and will meet up with each other more frequently. After the optimal temperature, however, the activity of the enzyme starts to decline because the enzyme starts to denature and lose its shape. If the enzyme loses its shape, its active site will no longer fit with the substrate and it cannot catalyze the reaction. In number 6, it asks which temperature is optimal for the enzyme whose reaction rate is pictured to the left. So that's referring to this graph right here. And notice that the rate of reaction on my y-axis is highest when my temperature is 40 degrees. I assume they mean 40 degrees Celsius. With pH, the acidity or basicity of the environment that the enzyme is in, each enzyme is going to have an optimal pH, just like they had an optimal temperature. For pepsin, it appears that the optimal pH is around 1 to 2. Pepsin is an enzyme that's found in your stomach, and the acidity of your stomach makes its pH around 1 to 2. Perfect for pepsin. Anything below 1 to 2 or above 1 to 2 is going to denature the enzyme, which means their rate of reaction would go down. Trypsin is another enzyme. It's found in your intestine, and its optimal pH is around 8 since the environment of the intestine has a pH of 8. So again, anything above or below that optimal pH will denature the enzyme. The reason that enzymes are going to denature at different pHs is because it causes them to unwind and lose their tertiary structure. It will break the hydrogen ionic bonds, hydrophobic interactions that exists between R groups that give us the tertiary structure. However, it's not going to break the covalent peptide bonds that occur between adjacent amino acids. So it won't break down the primary structure of a protein, but it will break down the tertiary structure. Another factor that could affect the rate of reaction would be the substrate concentration. If you add more substrate molecules, usually that's going to increase the rate of reaction. If there's more substrate molecules for the enzyme to act on, there will be more products formed. More products means you have a higher rate of reaction. However, if you reach a point where all of the active sites in the enzymes are being used and the enzymes are working as quickly as they can, you really cannot increase the rate of reaction by adding more substrate molecules. At that point, we say that all of the enzymes are saturated. So if you look at this graph here, it shows us the effect of increasing substrate concentration on the rate of reaction. As I add more substrate molecules, the rate of reaction increases until we get the point of saturation, where all of the enzyme's active sites are being used. At that point, you can still increase the rate of reaction, but you would have to add more enzyme molecules to do it. Okay, we'll skip through this. All right, this is just a math problem that shows us how to calculate the rate of reaction. You have an equation for rate on your formula sheet for the AP Bio test, so I just want to do one example of a rate problem. They tell you hydrogen peroxide is the substrate that's acted on by the enzyme catalase, and this is the enzyme that we're going to be using in our enzyme lab. And it tells us we're taking some data over 5 minutes, and we're measuring the amount of oxygen produced, oxygen being one of the products of the reaction, at each time in minutes. So they want to know what the rate of the enzyme reaction in milliliters per minute from 2 to 4 minutes. And they want us to round our answer to the nearest hundredth. So how are we going to do that? Well, this is a typical rise over run problem where we look at the change in the amount of oxygen produced over the change in time. So the way they calculated that was by subtracting the amount of oxygen produced at 4 minutes, or at 2 minutes from the amount produced at 4 minutes over the change in time, 4 minus 2. And we ended up with a value of 0.95 milliliters per minute. And on your actual AP test, you would have to grid in your answer. So they're just showing you how to do that there. Sometimes enzymes cannot work on their own. Sometimes they need little helper molecules that are going to assist them with doing their jobs. A molecule that is not organic that might assist with the reaction is called a cofactor. So that might be like a metal ion, like an ion of zinc, iron, or copper. And that's going to fit in a particular location with the enzyme and make it more receptive to a substrate molecule. Organic helpers are called coenzymes. So those would be like vitamins. Again, those are going to bind to a specific region of the enzyme and make them more willing to receive the substrate. Sometimes we have molecules that are going to prevent enzymes from doing their functions. And when they do that, that's called inhibition. We have two main types, competitive inhibitors or non-competitive inhibitors. Competitive inhibitors are going to actually compete with the substrate for the enzyme's active site. If they bind to the enzyme's active site, they completely prevent the substrate from entering. This is reversible. If the competitive inhibitor pops off, then the substrate can come back in. A non-competitive inhibitor does not compete with the substrate for the active site. It doesn't enter the active site at all. Instead, it binds to a different region called the allosteric region, and it causes a shape change in the enzyme, which makes the active site change shape and makes it unable to bind the substrate. Non-competitive inhibition is a form of allosteric regulation. Allosteric regulation involves any molecule called a regulator that binds to a different site on the enzyme, not the active site. And as I said before, we call that different site an allosteric site. We've got two types: the inhibitors, the non-competitive inhibitors that we mentioned before, and activators. Activators bind to the allosteric site and they make the active site more receptive to substrate molecules coming in. Okay, so I believe this right here shows us allosteric activator versus inhibitor molecules, and it shows us what happens when an activator comes in when we have an active enzyme form, and it's going to stabilize the enzyme in that active form. Now, normally the enzyme oscillates or changes between its active form right here, where it has a substrate that can, or an active site that can bind the substrate, and it also has an inactive form. Naturally, it will oscillate between these two forms. When you add the activator, it's stabilized in the active form. If you add the inhibitor, it's going to be stabilized in the inactive form with active sites that are not receptive to substrates coming in. Another example of the regulation of enzyme function is cooperativity. If you have an enzyme that has several active sites in it, a substrate binding to one of the active sites could trigger the rest of the active sites to be more receptive to a substrate molecule. So here they show us an enzyme in its inactive form, and once one substrate molecule binds, all the other subunits of that enzyme appear to be in the active form. It's not really a type of allosteric activation, since it would have to be a molecule that's binding to a different site, not the active site. You've got a question number 8 that says, which of the following characteristics is not associated with allosteric regulation of an enzyme's activity? And here it says a mimic of the substrate competes for the active site for option A. B, a naturally occurring molecule stabilizes a catalytically active conformation. C, regulatory molecules bind to a site remote from the active site. D, inhibitors and activators may compete with one another. Or E, the enzyme usually has a quaternary structure. I think by that they mean that it has multiple active sites, which we do see, since there are multiple polypeptides within that large protein. I'm going to go with A, a mimic of the substrate competes for the active site, because when you have allosteric regulation, your activator or inhibitor molecule is going to bind to a site that's different from the active site, called the allosteric site. Okay. In a reaction or a series of reactions that involve enzymes, sometimes the product of the reactions will inhibit one of the enzymes, so the reaction doesn't continue to occur. Sometimes the product of the reaction will stimulate the enzyme so that even more product is made. Negative feedback or feedback inhibition is when we have a lot of the product molecule, like in this multi-step reaction where we're trying to make product D. And that product, since we don't need to have any more of it, is going to inhibit an enzyme early in the process so that we don't waste a lot of resources making something that we already have, like product D. Positive feedback is the opposite. That occurs when the product of the multi-step reaction, like Z shown here, is going to activate the enzyme further so that it can make even more product molecule. This is a little bit less common than negative feedback because in general in your body you're trying to maintain homeostasis or stable internal conditions. So you always want to have a pretty constant level of a particular product, and if you have enough of it, you don't want to go over that, so you're going to inhibit the creation of that product. So this only happens, positive feedback only occurs in really specific scenarios. An example might be chemicals that are released by clotting agents called platelets that accumulate at an injury site. So if you get scratched and you start bleeding, platelets or certain types of red blood cells are going to accumulate and promote clotting of that blood so you won't continue to bleed. Those platelets will release chemicals that will then attract more platelets. So you want to get as many platelets as possible. All right, number 9 asks whether the situation that's given is an example of negative or positive feedback. It says, during labor, a uterine contraction causes the release of the hormone oxytocin. This, in turn, causes a nerve stimulus that stimulates the brain's hypothalamus to produce more oxytocin, which causes more contractions. This results in contractions increasing in amplitude and frequency. Is this an example of positive or negative feedback, and why? So here we're seeing the product of the uterine contractions, oxytocin, causing more contractions, which will cause more oxytocin production. So I would say that this is an example of positive feedback. The response, the creation of oxytocin, perpetuates the stimulus, the contractions. In negative feedback, the response stops the stimulus, whereas in positive feedback, the response encourages the stimulus. Last thing I want to say about enzymes is that within your cells, you have thousands and thousands of enzymes, and some of those enzymes have particular locations within the cell because they assist with a particular cell function that's localized. An example might be an enzyme that's involved in cellular respiration. That's usually going to be found within the inside of the mitochondria, the powerhouse of the cell, or in the membrane of the mitochondria. So that's an example of a localized enzyme. All right, guys, that's the end of part 2 of our Enzymes and Introduction to Metabolism lecture. And have a good day. Bye-bye.